So, hello everyone. People are already waiting for me. Hi, hello, and welcome. Microbehunter here. Uh, Finally, I am online now. I have to tell you, the last couple of minutes have been quite a bit stressful for me because I actually wanted to start this live stream showing you already a very nice tardigrade, but then it walked off my slide and I couldn't find it anymore. So that's a little bit of a again a demonstration effect starting right away, even before I actually got ready with the live stream. Okay, let me quickly put the cable away here. Okay, but still, I did prepare in case I'm not able to find any tardigrades. I mean, I found several of them today in the afternoon. I tried to prepare it, but right now they just walked off my slide. What we see here is a nematode worm, but I did prepare some videos if indeed I'm not able to find one. But I mean, I've got a whole bunch of moss here, so I'm quite confident. Well, first of all, again, hello around the world. I see that people already started to comment quite a bit here. Okay. And what you see here in the background is already a sample where I'm hopeful to be able to find a few tardigrades. I mean, there are tardigrades present. There are not many of them, I have to tell you. But I've seen already 5, 6, 7 of them today in the afternoon. But the one that I wanted to actually prepare simply walked off on me. Okay. So what I will do usually at the beginning is I'm going to send a hello all around the world now. I'm not going to read out all of the welcome messages here in the comments section. Okay. Yeah, and the sound seems to be good. Okay, here we are again. Okay. Yeah, there is already a question. Why is there a yellow tardigrade here on my microscope? So I'm going to quickly show you this one over here. Okay. Because this is a 3D printed model of a tardigrade, which I would like to share with you. I simply would like to show it to you, okay? Because it kind of illustrates a little bit how it actually looks like. Tardigrades are also known as moss piglets or water bears. And they are micro animals, so they're not single-celled, but they basically are made of multiple cells, and they have 8 legs. Okay, this is already quite a unique thing, and so when they first were discovered a couple hundred years ago, then people were not quite sure. What are they? Are they actually spiders? Because spiders also have 8 legs, right? I mean, they look very different to spiders, obviously. But they didn't really know how to classify them and what they actually were. And so they thought maybe they're kind of related to the spiders because of the number of legs. But in reality, they're completely a category completely on their own, right? I some time ago, I 3D printed this one over here because it kind of gives you a nice little illustration here. There's a mouse over here in front. They do look a little bit like aliens. So that's I think quite a nice thing, and that's why I put it here on my microscope as a little decoration here. Okay. So what I've got over here is a sample from a moss. I will show you then also how I'm actually I've collected the sample, and I have to tell you, I'm really a little bit very upset with myself because I really had this very nice tardigrade here under the microscope slide, really nice, and then it continued to walk off, and I basically did not pay attention for a couple of minutes, and then it was gone. Yeah, so we have to be a little bit lucky today. Yeah, if not, then I do have a couple of other water samples as well that I can look at, and yeah, we're gonna see how basically it's gonna take us. So if you want to find tardigrades, then I'm going to show you what I got here, okay, and how I like to collect them, and then I'm going to also show you the publication where it was first described. I collected a moss sample here. Basically what you have to make sure is that the moss, in many cases, it's quite dry, but you have to make it moist because the tardigrades are able to survive dryness. They're not able to survive heat very well. They don't like heat, but they can survive dryness. And when you make the moss wet and when you leave it wet for, I don't know, a day or two, then they basically come back alive, and then you're able to extract them from the moss, which I'm going to show you how I'm going to do that. Yeah. So where did I collect the moss? It depends a little bit where you live, but where I live is there is if you could just go into a forest, for example, where it's a little bit moist on rocks, you can find them quite yeah, in many places. So I collected this moss that was growing right outside my apartment. I simply grabbed it and it was quite dry, and then I added some water and 2 days ago, and yeah, I found tardigrades in here, but you have to be patient, as I essentially, yeah, just showed you. And then I'm going to show you now how you can actually, what you can do. You take a petri dish, or you can also take a plate if you want to, and you take a moss sample like this. It's all moist, and then you simply, yeah, you don't take the soil so much, but you can kind of dip it, yeah, like this with a lot of water, and then you're going to see that, yeah, there is going to be water collecting here on the bottom, with a lot of, let me move a little bit into the center here, yeah, with a little bit of sand as well, and some sediment. I want to, yeah, not to have too much of that. And then what I do is I kind of wait a little bit until the solid material starts to collect in the bottom of the petri dish, because many tardigrades, they like to cling to substances like sediment or leaves. Yeah, so they have little tiny claws, and they like to cling to a surface. Yeah. So it could be that they actually don't like to come off the moss either so easily. And yeah, so it's a little bit of a question of luck as well. And then what I do is I take a pipette and I simply collect a little bit of the solid material here, and I put it on a microscope slide. And what I usually do is yeah, as for a first quick look, I don't even put a cover glass on top yet. But I simply observe this under low magnification, simply to get an overview of what's present at all. Okay, so let me do that. Okay, yeah, here again is the microscope, and let's have a look here. Okay, I need to switch again, and that's basically what I'm able to see here. That's now low power. And yeah, at this low power, generally it's kind of difficult to see a little bit, but you can see some movement here. So I found a whole bunch of single-celled microorganisms as well. But we're going to switch over now to a slightly higher magnification. Okay, then what you have to do is you have to be really patient, and you have to scan the sample, hoping to find something. Okay, you can see that there is already quite a bit of life going on here. But yeah, it's a little bit of a question of patience now to actually find some tardigrades. And often it's not like this, that there are few tardigrades present, but sometimes it's like this, that they simply haven't come off the moss because they have little claws and they don't want to come off so easily. And this is basically what I've done in the afternoon. And I found a few tardigrades, and I put a cover glass on top, and I prepared everything for the live stream. And then I didn't pay it, and it started to move around on my microscope slide, and then I did not pay attention for a couple of minutes. Yeah. And then it walked off somewhere, and I couldn't find it anymore. And even though I was actually looking yeah, for it for, I don't know, yeah, 10-15 minutes before I started the live stream, I couldn't find it anymore. And it quickly prepared a couple of other samples, just like I've done now. Yeah, but they are essentially a challenge to find. And indeed, I'm not the only one who thinks so. Even the gentleman who discovered them found them rather difficult to find. Yeah, so this is basically what I've been doing right now. And yeah, and let's try the other water drop here on the other side here. Yeah. So maybe I'm going to go down with the magnification again to get a, but I have to be honest with you, I do not want to simply spend now the next hour or so just looking for tardigrades, and then it's also not going to be very interesting for you. But usually what's a good approach is always to look for a little bit of movement, okay? Like there's some movement over here, but this looks a little bit more like a rotifer. Sometimes the tardigrades, what they like to do is they also like to hide behind sediments. So sometimes if there's a little piece of sand or something like that, then sometimes it clings to the sand, and then it's kind of difficult to see. Okay, but there is quite a bit of movement going on here. So let's go up again with the magnification here. Okay, lots of sand. Yeah, and refocus again. So again, looks a little bit more like a rotifer and not like a tardigrade. Yeah. And this one over here seems to be like a ciliate, a single-celled ciliate. Ciliates are, of course, always single-celled. So I just would like to say that this is indeed a little bit of a hunt, and which requires patience. They're easier to find than face mites. Could this be one? I don't know. Okay, so I've seen something here which is kind of slow moving. So, but I don't know. Let me have a look. I have to be very careful now because I'm not using a cover glass. But this could be one. I don't know. I'm gonna use the arrow over here. Look, I've got this arrow. I'm thinking of this guy here, which was moving, not moving anymore. Okay. Yep. No, now it's moving again. Yeah. Yeah. In any case, you get the idea. Yeah. And I'm going to, tune down a little bit the light intensity again, and we can continue to scan. And as I'm scanning the slide, I'm also trying to keep an eye on some of the comments here. Yeah, so there are rotifers over here. Okay. For me, tardigrades are amazing creatures, especially under an electron microscope. They look scary under an electron microscope. If you just Google a little bit, then you're able to find lots of these images. Yeah, and I like to find tardigrades and observe them as they survive, as they revive out of the tunnel state. And I'm going to show you a video of a reviving tardigrade. So I did prepare that as an emergency, so to say, backup, because I was kind of worried already that something like this might happen, which is really, typical. I mean, I've prepared everything and it was beautiful tardigrade, and the guy simply walked off on, they couldn't find it anymore. Do you need to put some food on the moss sample like you do for the paramecium? No, not at all. So if you've got tardigrades, they find so much food on the moss because there's so many bacteria growing on there. There is I think, no point in adding extra food, right? So I think this is not necessary. So this one over here, that's again, look, seems to be a single-celled a protist over here swimming around. But no tardigrades. Here, there's another one over here. You see that here, because I made the moss sample moist, this also caused some of the other microorganisms to regenerate here. Yeah. Maybe the light microscope looks at it top down. It does not really look 3D, but still sometimes the cover slip actually squashes the sample a bit. That is correct. However, because there is usually some moss or some sediment on the glass slide, this keeps a safe distance. So I've not, up to this point, I could not, luckily, squash any tardigrades because there was always too much other material on the microscope slide, which kind of prevented the cover glass from crushing it. Yeah. I just saw a piece of microplastics. Yeah, maybe microplastic fibers or so. Unfortunately, I don't know where it is now. You find a lot of them. Okay, even if I collected some water samples from a small stream and from a sediment, you find microplastics. Yeah, so usually sometimes these are fibers. Yeah, that's of course a nematode worm here. This is a pretty big one actually. Yeah, you find them also quite often. Yeah. Why is it that you can see a 3/4 a side view image of an object on an electron, but you can see only 2D image under the light microscope? I'm not talking about stereo microscope. Yeah. What you're talking about is asking about is the following. There are so-called scanning electron microscopes, and those scanning electron microscopes are able to produce an image of the surface. Okay. There are also so-called transmission electron microscopes, which produce a 2D image. And what we have over here is when we look using a light microscope like we have over here, because the light basically passes through the specimen. For this reason, you basically only see the two-dimensional projection. Now, if you have so-called DIC, a differential interference contrast microscope, then some of the objects will appear to be a little bit three-dimensional because they're casting, it appears to be like they're casting a shadow. Yeah? So what I can do, for example, right now, I'll remove the filter here, and then you're going to see that this is a regular bright field, and it looks a little bit more flat as well. So this is probably the way that most of you are going to see the image, because it's in bright field, but I've got this little thing here which gives it a little bit more color here as well. Okay. Do you use regular tap water to put on the moss? Yes, I just use regular tap water. This is perfectly fine, unless it's chlorinated or there's some other stuff in here. That's perfectly fine because there are so many impurities in the moss and in the soil of the moss, so that essentially it's not going to be an osmotic problem. Yeah, but distilled water I probably would not use, right? Because there are really too few minerals. But I would say that that essentially regular tap water works. So what I'm going to do now is I'm going to, sometimes if there is a very slow movement, but this is too small. Yeah, sometimes if there's a slow movement, then it could be something. But I did prepare a video, just as an emergency backup. But this is really upsetting because yesterday when I actually put the moss sample on the slide, I could see 3 or 4 tardigrades right next to each other. It's really surprising, right? And I said, wow, there are plenty of them in the moss sample. And in the meantime, I can't find any, and I'm kind of wondering if they have not moved away into, I don't know, a certain direction, maybe towards or away from light or something like that. Or deeper down into the moss. That would be also a possibility. Yeah? Do you mean that the tardigrades found in the residential area is different than in the forest or park? Why not? What I read is there are 1,300 different tardigrade species. So chances are pretty good that wherever you find in different areas, different moss, you're going to find different tardigrades. Yeah, that's quite well possible. But simply because there's so many species around. Yeah. I asked you before if tardigrades have a voice. What do you mean voice? If they're able to speak? Probably not, because why would they? Usually microorganisms, I don't know if they're able to communicate maybe over vibrations or something like that. But by making that's possible, but certainly no voice for speaking like we humans have, because but it wouldn't surprise me if certain micro animals or certain animals are able to communicate over vibrations, for example. This wouldn't surprise me. In that sense, you could actually say that these are also kind of like sound waves, but this is of course not something that can be compared to human voice. Maybe it depends on the place, as long as it's not prone to extreme heat, which would completely dry the moss. As a matter of fact, if the moss dries completely, you can revive the tardigrades by making the moss wet for some time. But what I read is while dryness is okay, what they don't like is they do not like heat. Okay, so if it really becomes very hot in the summer, then this is probably not a good idea. Yeah. How many megapixel is your microscope camera? I'm recording. My camera is able to record 4K, but this live stream right now is 1080p. And I don't know how many the photo camera, how many megapixels it has, but you see the megapixels really are not really the thing that are so important. Yeah, I'm really getting a little bit upset now that I'm not able to find any. Okay. The megapixels are really not so much of an issue because usually the camera have many more megapixels than what the microscope is able to resolve. So generally, there are quite a few of them. So maybe I'm just gonna go off to the side here a little bit, and maybe I'm going to try to find something over there as well. What makes tardigrades so tough? I don't know about that, but it's nothing unusual for a certain organisms, microorganisms, to have a dry resistance. So that's their mode of survival, because when they live in moss, they have to be able to survive also dryness, because moss is not always in wet. When it doesn't rain and when it dries up, then the tardigrades also have to have some kind of a possibility to survive. And this is really a little bit unusual, because honestly, I've didn't have any problems yesterday when I tried this. Just on the first try, I already saw 3 or 4 of them. Going to go over to the other drop of water that I have here. Let's try to have a look here again. Okay. I still don't understand. Are you talking about the survival in different environments or different types of tardigrades in different environments? Well, there can be, of course, different types of tardigrades in different environments. That's actually what's normal in nature. Yeah. Not all the moss species are the same, obviously. So very much enjoy your channel. It helps me with my own microscopy. I like it. Thank you very much that you enjoy the channel, even if I'm not always very successful. But this has also become maybe a little bit, I mean, I got the feedback from some of my viewers. They say that this is actually more realistic as a form of microscopy, that essentially you're not always able to find the things that you want to look at right away. Because in my YouTube channel, of course, I'm only able to show you the things. Are we lucky now? Just a second. I think we've got one. Without a cover glass, so I'm not able. I'm very happy. Finally. Whew, I'm relieved. Here's one. Without cover glass, okay? So that's really important. I cannot go higher with the magnification, otherwise I'm going to dip the objective into the water. And that's why it's a little bit blurry as well. Hooray! Success! Okay, now the evening now the evening has been saved. I say evening because where I am, it's evening right now. Okay, finally. Okay, so that's yeah. It's called fishing, not catching. Yes. So if I now put a cover glass on top here to make it a little bit flatter, so then there is of course again the danger that I'm going to lose it out of sight. Okay, but I could go up higher with the magnification though, right? So this is we've got one. So, and I want to talk a little bit about those tardigrades now. Now that we've got one, this is now I'm using now the 20 times objective. Okay, just for your information, and essentially, that's actually one of the reasons also why they might be a little bit difficult to find, because they like to be around solid material. As I told you already, they like to cling to objects, and they don't move quite as rapidly as some other organisms do. The reason is on a smooth microscope slide, they have a real problem crawling forward. I've once managed to completely isolate a tardigrade, so without any other sediment or solid material around. And then it was always slipping. So it was kind of walking, but it was not able to move away too quickly, only very slowly. Yeah, here you can kind of see that it's kind of clumsy a little bit, yeah, as it's trying to move forward. Yeah. Yeah. Do only some tardigrades have eyes? Well, I mean, let's put it this way. Many of these microscopic animals and so are light sensitive, but this does not mean that they actually have eyes, like we are able to like we have eyes. So in other words, correct me if I'm wrong. I think tardigrades don't have eyes as such, because otherwise you would see the eyes as dark spots. Because if you want to absorb light, then there has to be a pigment there, and then it must be darker. But I've not seen any dark spots on a tardigrade that could resemble eyes. Yeah, but still, maybe they are still light sensitive, that they actually might either move towards or avoid light. So this is quite possible. Okay. Yeah, so I'm going to go through again some of the, as we have a look at this one over here, and then I'm going to show you the short video. Some tardigrade types are in the ocean too. Yep, that's what I was talking about, heat. Okay. Yeah, I'm talking about survival, even though they are pretty tough on heat, but they don't like heat though. At least that's what I read somewhere. Okay, then we found the tardigrade. So there is some communication going on in the chat, which is fine. So DSC optics can give the appearance of Reinberg illumination. That's an interesting question from Micro Addict. DIC actually gives it can imitate Reinberg in the sense that their background has a different color than the specimen. And if I rotate or shift a prism, then you're able to see that I can even change the background color. But actually, the technique is very different. And essentially, what the DIC does is that it gives, because of a differences of refractive index, it gives some objects the appearance as if they are three-dimensional, as if a shadow is being cast. However, this does not resemble the actual 3D appearance of the object. So for example, if you look at some of the sand grains seem to be a little bit darker on the one side and then on the other. So they appear to be a little bit having some kind of a depth to it, right? And that is what DIC actually does. Yeah. So it uses polarized light and all these things. And the reason why I've decided to get, look, here, a protozoan, to get DIC is simply when you make YouTube videos and live streams, it's simply nicer to see a little bit of color. Okay. And also interesting here, in the tardigrade, you're also able to sometimes look into the organs that they have. Now, if you were to zoom in much more, which I cannot do now because I don't have a cover glass on top here, it's risky, okay, then you could actually see also the individual claws. You know what I'm going to do now is just, I'm going to read a little bit through the comments again, and then I'm going to talk a little bit, I'm going to show you a video, okay, where I've actually dried a tardigrade and brought it back to life, okay. So let's have a look here. So lots of conversation going on here. Is the tardigrade eating something or something else is trying to eat it? It's difficult to see what's going on. Let's put it this way. The tardigrade is always probably eating something if it finds bacteria or something, but it's trying to crawl ahead, and it's not able to move forward because there is nothing to cling to. Okay, so that's usually, it's kind of like walking, I almost want to say like walking in air, walking in water, yeah. But because everything is so smooth, it's not able to move forward. Yeah. So actually, what I tried to do as well today afternoon is I tried to actually get using my pipette here to isolate some tardigrades, at least the few that I found. from the other solid material, because I wanted to get rid of all of the sand and the solid material, which was very difficult to do because it was always clinging to this, to the sediment that, it didn't want to separate it, with its hind legs. So with the front legs, it was always looking for something to hold on to, but with the hind legs, like right now, it was always kind of clinging to some solid material. Is a tardigrade a herbivore? Not necessarily. They eat all sorts, and they're even cannibals. Some of them I read. So at least, I mean, after all, there are 1,300 species, I guess they're not very selective. As long as is it small enough to be taken in over the mouth? Bacteria, for example, that's not on the side. That was a rotifer, kind of walking down here. No, it's gone. Yeah. So it kind of, they basically, tardigrades, they have these things. They don't have these teeth or so, but they have these things kind of to suck up the thing. I've tried to take a sample from the beach nearby I live, but I have not found them on the sand samples yet. I don't think that you're able to find them so easily in the sand itself. Okay, tardigrades essentially, being called moss piglets, can be found a lot in moss. I'm not saying only. I found some of them also in lichens. Here. So what I'm going to do now is the following. I'm going to first show you now a video that I made. Here we go. And that is a video now. That's a tardigrade. I don't know. I think it's now on a, I don't know what the large round thing is here. Okay, let's this large round, where's the arrow? So this large round thing, I don't know what it is. Okay, but the tardigrade here, as you can see, really likes to cling to the object, and essentially, I waited for the water to evaporate. So we're gonna have a look at that. I was kind of focusing again, and there's now a very small water drop, and the water evaporates more and more and more and more. The tardigrade starts to become smaller and smaller and smaller. Okay. And you can maybe also see that because the water droplet kind of starts to pull together around the tardigrade. You see now it starts to dry up. Okay, and it's dry, or almost dry. Okay, so that's basically what I've done first, and then of course I tried to add some water, and I tried to revive it again after a couple of minutes. See, it completely dries up. Okay, so that's basically what I ended up with. And now in the second video, I'm going to add a little bit of water. And let's see what happens. Okay, so see, from the bottom right side, there is hidden behind my face a little bit. There is, from the bottom right side, there is now some water approaching. And here is the water is now approaching, and it's now contacting the tardigrade and whatever it was onto. And look, it kind of starts to swell up again. It soaks up the water again. And the question is, of course, now, okay, it starts to become large again. But the question is did it survive that ordeal? And then you have to wait a little bit, a couple of minutes. And as a matter of fact, of course, it came back to life. Now look, it's slowly starting to move. Slowly starting to move. And the legs are also then slowly extending. And then after a couple of minutes, it's back to normal again. So, by the way, this is not a video that I recently made. This is actually a slightly older video in my main channel. In the other channel, if you search for tardigrades, then I think I called it my tiny little water bear or something like that. I actually documented this whole process. So this is not time lapse now, okay? But in the other video in my other channel, I did actually make it in time lapse. Let's have a look here. Yeah, it's still still 3 minutes to go. If you want, I can fast forward. Look, if I fast forward the whole thing, I'm fast forwarding the video now. And now back to You see, now it's starting to move again. I consider it quite remarkable, honestly, to be able to see that. Yeah. And of course, you've probably already heard that they sent them also to space. They even sent them on to the moon. Yeah. Yeah. So, and I think maybe one of the reasons why they're so yeah so well liked or beloved is because they're not so easy to, as you've seen, they're not so easy to find. Yeah. So you do have to be a little bit patient. I mean, I started to say before that the face mites are, yeah, it takes you even more patience to find those. At least if you watched my video where I put a tape over my forehead to collect my face mites, this it does require even more patience. So maybe you do have to check for a couple of hours until you find one of them. But with the tardigrades, I mean, yeah, generally you shouldn't have any problems. But as you've seen now, yeah, you might spend maybe 20 minutes, 30 minutes looking. Yeah. Okay, here the video starts to repeat. Yeah. I found a dead tardigrade a while back, and the two stylids had relaxed and were sticking out of the mouth. Yeah, very easy to see. I've also seen not only dead tardigrades, but they like to molt, so they shed off their skin. And then basically it can also be, okay, here it is still. Yeah, and it can be that you have maybe also seen some of the empty, yeah, the empty skin of some of the tardigrades. Okay. Would you try the same hydration and rehydration process for a week to see if it comes back to life? Your sample might need to be refrigerated. Yeah, I could try that. What I would like to do in this case is I have to kind of find a better way of finding more of these tardigrades, because you see, I've been searching now for 10 minutes or so until I found this one over here. And then I would like to try to isolate them using my pipette a little bit better. So then I can dry them, and then, yeah, we can do all sorts of experiments with them, right? How long do tardigrades live? Because they said they live forever. Well, I don't know if you can say that they live forever, but I mean, they are very resilient, and they are able to withstand cold and also dryness quite well. In that sense, they have been studied quite a bit, also by scientists, because yeah. But if they have, I don't know what they're. Yeah, if they are able to die of old age, so to say. Huh? Yeah. Today in the afternoon, I've also seen, I don't have the video right now, also tardigrade eggs I've seen, which quite nice as well. Huh? So I would like to show you now. Okay, look, what do we have down here? Here, that's a ciliate again. Could, I don't know, it looks a little bit, no, it's not paramecium, yeah, but here, the single-celled protist. Kind of also shows you a little bit the size relationship, that this protist down here is made of one cell, and the tardigrade just a bit larger, yeah, right next to each other right now, just a bit larger, made of maybe around 1,000 cells. So it's also quite, yeah, remarkable of how cell size can be different. Let's see now the protist and the tardigrade next to each other. Change it a little bit to blue. Up again, it's a little out of focus. Yeah. I learned that some microorganisms appear to express fluorescence proteins that they can be detected within specific excitation emission wavelengths under fluorescence scope. Let's put it this way. I mean, I mean, I don't have a fluorescent microscope, but this is nothing, that would surprise me either, because there are, of course, naturally, it's called autofluorescence, where essentially certain chemicals are able to shine in UV light. So yeah, but unfortunately, I do not have a fluorescent microscope. Yeah. Is it better to observe tardigrades without a cover glass? No. And that was my mistake, maybe. It's actually better to use a cover glass it's almost always better to use a cover glass. The reason why I did not use a cover glass now is because I wanted to quickly check, and I was a little too lazy. I'll be honest with you. I was a little bit too lazy to yeah, to put a cover glass on it, simply wanted to check. And then I found this one over here. And now if I put a cover glass on it, I might have problems finding it again. Yeah. So this is a little bit the issue that I'm having right now. Yeah, I would actually like to go up higher in the magnification, but I can't. Yeah. So this is now, yeah, with the 10 times, and this now is with the 4 times objective. Yeah. Huh. Yeah, and you can see that with the 4 times objective, it's already kind of difficult to actually find them. But what I could try, I mean, I don't know. Now that, no, oh, where is it? Here it is again. Yeah. Yeah, if I put a cover glass on top, then everything's going. I don't know if I should try that. You know what? I'll try towards maybe the end of the live stream, so that people who join in still are able to see at least something here. Okay, yeah. So let's leave it here like that. Okay. Yeah, it's cool that tardigrades and rotifers at around 1,000 cells each can be about the same size as some ciliates. Yeah, I also consider this kind of fascinating. The cover slip inhibits the movement a bit, and the tardigrade won't be so out of focus. Yes. If you want to move it freely and you don't mind the poor focus, just use a cover slip. Yeah, so generally, because there are so many sand grains in there, I probably do not think that the cover slip is going to crush the tardigrade, because the sand grains lift up the cover slip a little bit. Fun fact, just as rotifers, they have utili, yes, which means that the individuals inside some species have exactly the same number of cells. I tried to, or somebody actually sent me a link to that. Yeah, somebody who actually found out that the cell size is that the cell number is the same. Apparently, he did some staining techniques and he was able to somehow count the cells this way. Yeah. So that is yeah, quite interesting. You know what I'm going to do now is I'm going to show you now a PDF here. Look at this. This is written in German, and there is a link. Actually, over the Wikipedia article, I had this link. And where is the? No, that's not the one. Here it is. Okay. Yeah, I'm now able to. Yeah. And look, this is written in very old writing, but this is the first publication of and the first description of tardigrades. It's written in German. Even I have to concentrate a little bit, but the text is actually really fun to read. It's written in a very unscientific way, first person, but the author, who has described tardigrades, essentially said that it's a very strange and a very rare animal. And he said it's very rare because he's found it in winter only once, and in summer he hasn't found it at all. Maybe because the moss was dry. I don't know. Yeah. And then he talks here about the body shape. Yeah. And then he said it looks like a little bear, but you should not be afraid of it. Yeah, and it's worth looking at it. So it's a kind of written in a very colloquial way, actually a very fun way. Yeah, and here, for example, he says you should not be afraid of it. Yeah, of this bear because it's so small or whatever. Yeah, yeah. So it's just I just wanted to show you that this is actually the first publication where it was described. And if you go all the way down, this is actually a huge book. Because the guy who described this actually described a lot of things. I think there's somewhere at the bottom, there's some kind of a drawing even. Yeah. And he actually sent, and that's why I call it the little bear, a little water bear. So basically he has given it a name. I wonder where the drawings are. Somewhere at the end. Here. Look, here we go. So that's basically the drawing that he made of this water bear. He counted the legs, obviously, as well. Yeah, but why the 2, 1, 2, 3, 4? Why are there only 6 legs? There should be 8. Yeah, but yes, but yeah, seems to be some kind of larvae. Yeah, so just wanted to show this publication to you as well. because I found it kind of interesting. I like also a little bit the history of science. So usually what I do is want to know a little bit more, like water bears, moss piglets or so. I go into Wikipedia. Sometimes there's some references, and sometimes even the original source is cited. And then if the work is kind of old, then sometimes in a library you can find the PDF because it has been scanned. Yeah. Yes, that's correct. Johann August Gose, that is basically the guy who discovered it. Yeah. So actually, I got it over the Wikipedia article. Okay. Yeah. So again, a couple of comments here. How do you clean the cover slips? I have tried several makes and they all appear very dirty. If the cover glasses are dirty, then what I do is where is it? I don't have it. Yeah, here it is. This is a microfiber cloth for cleaning glasses. So this kind of works well. And what I also found to work well is sometimes it's not really dirt, but it's some kind of they're called water spots, calcium carbonate. So a little bit of vinegar also helps because it dissolves the calcium carbonate. And if you want to really clean the slides, then I find it quite good if you use dishwasher. Just show it to you. Look what I got here. I'm going to switch over to the desk. So these are all the box of used slides, and I simply collect them and then I clean them. Yeah, just to save a little bit glass a little bit. Yeah, here we still have the water beer here. I'm almost a little bit afraid of moving away here. Just use soapy water to clean my slides. Yes, soapy water, hot water also works. Just make sure that if you use soap or any detergent, that you really rinse it very well. That's important because even small amounts of soap can actually harm the microorganisms. Yeah, because it's a detergent and it dissolves the membrane. So I remember when we were in the lab, we were not allowed to put our equipment, our lab equipment. We were not allowed. We had 2 dishwashers. And we were only allowed to use 1 dishwasher, and I think there was not even any dishwashing powder added to it, but it was only rinsed with hot water, if I remember correctly. And the other one was used for our plate and so on. Because if you added some dishwashing powder, it was very difficult to remove the small amount of detergent even. Yeah, so we were kind of told that we had to be very careful about using soap in the lab. Of course, we were doing research, so it was a little bit different, because they told us that it's very difficult to sometimes remove the small amount. See, that's always a little bit unfortunate that it's always kind of hiding a little bit behind some material. Google should make a whole library of microorganism emojis. Yeah. I took a sample of water from a big puddle that formed in my field from a snow melting, caught a mosquito larvae in it. Yes. Generally, there is the saying that a puddle should be a few days old. And if a puddle is a few days old, generally you're quite successful in finding something interesting. And so, talking about deer, have you run into the situation that you cannot completely remove dust on lenses that they are in the light path of the microscope? This can happen. So basically, well, let's put it this way. It is possible for dust or dirt to be in the light path. In many cases, or in most cases, you won't be able to see the dust because it's out of focus. The most problematic, or one of the most problematic places of dust is directly on the camera sensor. So that's because this is where it really casts a very strong shadow and it makes a strong speck. But sometimes, in most cases, the dust is out of focus. Sometimes, if it's really disturbing, I had a very on my other microscope a very disturbing piece of dust directly on the optics of my lamp. So I had to take apart the microscope and clean the optics of the illumination system because there was a really bad speck there that really disturbed. And what I suggest is that if you want to find out where the dirt is then try to rotate the different parts. Rotate the eyepieces and see if the dust or the dirt also rotates, simply so that you can narrow down where you're able to find the dirt. So, is drinking water different from tap water as seen through a microscope? I don't understand the question in the sense, well, it generally, I don't understand the question because where I live, drinking water and tap water is the same thing. So we actually have tap water and we're able to drink it. But generally, if you just take pure tap water, either out of water out of the bottle, and put it directly under the microscope, it doesn't matter. You're not going to see anything. Because generally drinking water and even tap water, even if it's not for drinking, generally is so clean, still too clean to be for you to see microorganisms. So normally what they do, and I've not done this myself, but just remember this from university, if you want to actually check the water quality of tap water, you actually have to concentrate it by passing the water through a filter and then observing the filter. Otherwise, the concentration of microorganisms might be too low. Yeah. Does the pH level have a great effect on microbes? Oh, yes. Oh, yes. Of course, it always depends on to what extent. Different microorganisms have a different pH tolerance, but generally, yes. So if you go beyond the limit, then you're going to kill them. Yeah. Yep. If it exists, yeah, I clean the glass. Okay. Yes. Again, some communication. What is the difference between tardigrades and tardigrades? Oh, that's an interesting one. I don't know, but that is something maybe someone can research. The true tardigrades and tardigrades. Interesting. Yeah. I use spray air as needed for trouble specks of dust. Be careful with spray with air in a compressed container on optical parts. You can use compressed air without any how do you say, just regular compressed air. And what I've made myself is using a plastic bottle and a bicycle pump. It's possible to make a little compressed air container. But if possible, don't use those commercial spray cans for blowing dust away, because they contain some kind of a propellant. And when it expands, and it cools down very quickly, and you don't know if there's some kind of deposits forming. Those spray cans are good for removing dust and dirt from computer parts, for example, but you probably should not be using them on optical surfaces. But it's okay to use them on other parts of the microscope, to remove the dust. But I would probably not risk it on the optical surfaces. Drink tap water. Yes. Yeah, of course. The tap water where I'm living is drinking water quality. I only drink tap water, without boiling even. And this basically means that the tap water is so clean and contains so few microorganisms that you're not going to see anything under the microscope. Yeah. I often do a final clean with deionized water, distilled water. Yes, the reason why sometimes you want to do a final clean of the slides with deionized water or distilled water is because you want to remove the water spots. Water spots are calcium carbonate deposits, and those deposits can be a little bit disturbing, and either you dry wipe it or you rinse it in distilled water, and then you also do not have those water spots. What about a brush? The ones for glass surfaces, that's not a... where's my brush? Look what I got here. Okay. Yes. As a matter of fact, I use this brush here, but I do not use it directly on the microscope objective, right? Because there is no need. It should not become dirty at all anyway, because the microscope objective points downwards, so it does not collect any dust, and it should not get into contact with the liquid anyway. Yeah. Yeah. So what I did not know that was possible. I guess this refers to drinking water and tap water being. Where are you again? Well, I'm in Central Europe. In many countries in Europe, luckily we have good drinking water from the tap. Yeah? Okay. I know what you mean with a canned ear, but I had never had issues with for many years. Oh, that's good then. Well, I guess it's because maybe the propellant evaporates immediately, and therefore there are no deposits. This could be the reason. Yeah? Austria. I'm from Austria. Okay. Yeah, I'm going to get some fresh pond water with some of the mud on the bottom. Yes, the mud is important, and as a matter of fact, today I collected some, but not so interesting, unfortunately. Yeah? So the tardigrade is moving vigorously. I'll be careful not to lose it. Yep. Yeah, so I will simply try the following now. I'll try to remember where it is. Yeah, here it is. But you know what I could try is I could try to... Yeah. You know what? I'm going to try to remember where it is somehow, and so that I'm going to find it again. I would like to try another sample in any case. So I have to remember it somewhere. Yeah. Yeah, here it is. Yeah. You know what? I'll just leave it as it is right now. Okay. Because if I find another one, it's going to look the same anyway. So it might not be worth searching for another one, okay? Because then it's going to look exactly the same anyway, and then, yeah. But for those of you who have joined in late, if you have joined in late, I just wanted to show you again what I've done here. This is a moss sample that I collected 2 days ago from right outside. And I added some water so that basically the moss starts to regenerate a little bit, becomes a little bit more green. And I simply have taken, not from the bottom side so much where there is, it's muddy, but actually from the top side. Okay, and what I've done is I simply dipped it here and tried to squeeze out a little bit of the water. Yeah, and then I've taken a pipette here and tried to take up some of this and put it under the microscope. And if you're lucky, you're going to find some tardigrades this way. Again, only those that are able to come off, because most of them, as I told you already, they kind of like to cling to surfaces. So so that's simply a thing. Or another thing that you can do is I've got a little water bottle here. You can try to rinse it down like this. Okay. And then you keep it tilted, and then you wait for the sediment to settle a little bit, and with it also the tardigrades and whatever other microbes, they might settle down a little bit, and then you can collect some of that from the sediment and put it under the microscope. Okay, now we know a little bit more about tardigrades, or a class of phylum of tardigrades. They have some morphological peculiarities and mostly are adapted to freshwater environments. Thank you for the information. Is it it is painful process to determine the exact location of dust in the light path. Cleaning it is another pain. Air removal might not work to the charges on glass. Yeah, so basically if there is some kind of an electrical charge, then it's possible that it tries statically to attract the dust. I found the most of the dirt actually starts to accumulate obviously on the eyepieces, because when you are close, then the eyelashes, they will touch the surface of the eyepiece, and then this is a little bit greasy, and then of course the dust also likes to stick. So generally I recommend that you, and I try to do that, to always cover the microscope and to try to minimize dust as much as possible. But you know what, I'm going to just show you again over here on the bottom. Yeah. You might not be able to see it here, but there is also a lot of dust over here. But because this dust is out of focus, it doesn't disturb a lot. Yeah, and for these things, I usually use always a kind of the brush. Yeah, you can actually see the dust flying around in the light path. Yeah. So this is, unfortunately, dust is difficult to prevent. Yeah. Yeah, but that is simply something that we have to live with. Yeah. You should use some Q-tips and some alcohol or some cleaning solutions specifically for glass surfaces. Yes, I occasionally do that. I use alcohol, pure alcohol, for cleaning away some grease, which accumulates on the surfaces of the eyepieces. And so far, I've not cleaned at all the microscope objectives, with the exception of the oil immersion objective, which I've simply carefully removed the immersion oil. Yeah. You need a tardigrade ring to keep it in field of view. I have a nice cleaning kit for my microscope. Yes. Do you reuse pipette tips? Yes and no. Okay, basically what I do is the following. These pipette, oh, I can show you here. These pipette tips are not sterile, and they don't have to be, because I'm not working I'm not growing microorganisms. So what I usually do is I have somewhere some water and I rinse it out, because what's the point of throwing it away when essentially tomorrow I'll be picking up the same sample again. But sometimes what I try to avoid, nevertheless, is I try to still avoid using this pipette tip now, and then going on over into another water jar, because I'm transferring microorganisms. Also not a big problem really, but it could be that there are, I don't know, some I've had this before, some insect larvae in here maybe, or eggs of insects, and they kind of start to hatch and then they eat the microbes of my other jar. So I kind of want to prevent these things. So what I usually do is if I'm using the same the pipette over on the same specimen, of course I reuse it. Sometimes if it becomes too dirty, then I simply will throw it away. And what I've got here is, I can show you, a box with pipette tips, and I bought myself also a bag of these pipettes. And because I removed the cover, the lid here, because it's easier this way and it doesn't have to be sterile either, right? So if you worked in a microbiology lab, then you know that this has to be always covered and sterile, but that's not what I need to do here, because I'm just doing microscopy work. Yeah, a mini vacuum cleaner to clean the slides. Difficult, because of what happens is that when it starts to dry up, then it starts to stick. The dirt starts to stick. Let's have again. Yeah, it's still here and happily moving. It's time for the videos. You said, these are the videos actually which I already showed you over here. Yeah, these ones over here are the videos. Yeah, and if you want to see yet more tardigrade videos, I can always refer you to my main channel. Okay, where I really zoomed in a lot, because in the other videos that I made, I really took quite a bit of time to isolate the tardigrades. So what I've done, and this was really a very painstaking and tedious process, I went with a pipette close to the tardigrade, and I tried to suck it up and then try to transfer it to a different slide, added fresh water to it to rinse away the sand. So I repeated this several times, and sometimes I even lost the tardigrade in the process. But then I actually got a much nicer video. Yeah. But this one here is a little bit more the authentic process here. Huh? For delicate optics like coated optics or telescope aluminized mirrors, there are pour and peel solutions. You don't wipe or scratch the surface. It works for big areas, but not for tiny optics. That's an interesting one. So basically, you've got a telescope mirror, and with maybe some dust sticking to the mirror, and maybe you pour something over it, it becomes solid, and then you kind of peel it off and it kind of like a glue, maybe. Yeah. What species or family are tardigrades in? To my knowledge, tardigrades are their own phylum. Okay, so that it means they're quite a large category. Are you gonna show pollen from last week? No, because I was not successful, unfortunately. I wanted to. I'm working on it. I'm trying to do pollen germination. So basically, this means where I add pollen, where it's some sugar solution to the pollen, and then you have a pollen tube forming. And I wanted to do a time lapse of this, but I was not yet quite successful with that. Yeah, so I have to find the right pollen. I think the tardigrades are fascinating, partly that I think that they are their own species. I got to tell you, they have 1,300 species. Yeah? So I think what you mean to say is that they're their own phylum. It's a much larger category. If you talk about species, then you're already quite specific. Okay, so there are over 1,000 species of tardigrades. Yeah? I think the tardigrades are fascinating. Yep. Yes, I did use Q-tips, wipe isopropanol, which is fine. Non-ammonia glass cleaning solution, none of them is helpful. Q-tips and wipe. It could be that maybe that you were trying to clean a place where there was no dust. Okay, so it could be that sometimes the dust is maybe in the optical path somewhere, and that therefore you were, even though you were trying to clean the surfaces, you were actually not cleaning the place where the dust actually was. Okay. Is it okay to use a microfiber cloth? Well, it depends what you want to clean. If you want to actually clean the actual objectives, you should not use a microfiber cloth either. You have to use lens paper, and you have to really be very careful. You should not apply any pressure when cleaning. Yeah, because as soon as you apply pressure to the optics, you might end up scratching the surface because there might be a small sand grain or dust grain, and then you might end up scratching the surface. So no pressure at all. Yeah. Yeah. So, yep. I have been waiting for this live stream for quite a while. Okay, yeah. I think where you're in the middle of the night. So, yeah. What I would like to do in the future, not in the future, actually, this is my plan, and I might actually document this as well. Maybe this could also be a nice, how do you say, a nice topic for live stream. Is I would like to make my own small, not aquarium, but terrarium, where I keep, maybe a plant or two, moss, and where I have a so-called a little, I've got a whole bunch of jars with water samples standing on the window, but I'd like to also have grow my moss a little bit there, so that I always have some moss samples. So this would also be quite decorative, and then you grow your moss in there. Maybe also possible to grow a few fungi in there, so that you're able to also always have a few more things to observe under the microscope, not just water samples, which are quite easy because you just scoop up some water sample from a jar with a jar, and as well. Yeah. The specific part of the scope is removed and put under the stereoscope. All dust can be viewed. Just addressed your recommendation. I hesitated to use it since it is more polishing on the surface. Yep, I think it refers to the use of lens paper. You may have already checked it, but on the top of the condenser lens below the slide is where I usually get dust trapped. So what I suggest is that you indeed ask yourself all the time, is it really worth cleaning? Because look, you will only notice that dust is present if you actually move the slide like I'm moving right now. And if there is somewhere a dark spot that doesn't move, then you actually notice that there is dust there. But if you do not move the slide, then the dust kind of blends in a little bit with the other things on the microscope slide. Yeah. Because there's actually, if you want to see it like this, there's actually much more dirt and dust on the sample over here, all of the sand grains and all these things, which basically, are there as well. But if you have now dust somewhere and it's not moving, then you actually don't see it because it kind of blends in with the background. Yeah? That's by the way also a trick sometimes to to make videos, even if it's dirty, then you just don't move the slide around, yeah, and then you don't see the dust. Yeah. Can we look at another sample? Okay, I would like to see more. Okay, you know what? Before I do that, I'm going to risk now something, because I've been now talking anyway over an hour now, so it's possible to risk now the following. I'm going to now add a cover glass to this. It's going to spread everything, and yeah, I might have problems finding the tardigrade again. Yeah, so I'm not going to spend a lot of time. I'm not going to spend a lot of time searching for it now. Okay, if I lose it. So I've got over here a cover glass, and I'm just going to dry wipe it a little bit. So, okay. I'm going to try to simply try to drop it somehow directly on the, look what I got here. I got this, special tweezers. The people who collect stamps also use those. You can actually use those. Great. I dropped it in any case. I've got tweezers, but then I'm dropping it in any case. So, just a second. Yeah, here we go. And just going to try to put it directly. The tardigrade is now right in the center, but it's going to be now flushed away. Up. Do I still have it there? Ooh, okay. It might still be there. It might still be there. It's a little bit out of focus now. And let's see, because yes, it's still here. That's great, because now I can really go up with the magnification. That's nice. Okay. So luckily, yeah, it's still there. So it's now 200 times. Go up with. Okay. So, and now let's really make it big. I'm very happy that this kind of worked. So, go up with light. Of course, the higher I go up with the magnification, oh, you see the water liquid is spreading under the cover glass. Okay. Okay, here we go. Always moving. Maybe if we're lucky, we're able to also see some of the claws. And I'm going to now try the following. I'm going to go up yet higher. 600 times. I have to adjust the condenser as well. Go up with the light intensity, open it a little bit to give it a little more light. That's now 600 times. You see that not so good. Maybe you see that it's already a little bit, yeah. The depth of field is quite low. Still too much water, still too thick. Let's try like this. Give it a little more light. Yeah, you see, large magnification is not always the best. So let's go down to 400 again. So let's give it a slightly more neutral color for a change. And then we're also able to see the digestive system a little bit here. Okay. Yeah, so that's now 400 times. So, so dust within the diaphragm. Tardigrades might be resilient. Yes, I didn't get. I put a bit of Vaseline on each corner of the cover glass not to crush the organisms. Yes, that's also a very common way of actually doing it. You can use Vaseline, or you can also use Play-Doh or plastic. How do you call this? Yeah, for kids to make things. Yeah, you can also use them to make little feet to kind of adjust the height of the cover glass this way. Are tardigrades found in the subtropics and tropics? I assume with so many species. I'm quite sure that there where there is moisture and water, I'm quite sure that you'll find them over there. Yeah. Most of the dust are out of focus. I will give it one more try and move on. Yep. I'm afraid if you use too high magnification, it will be out of focus and you have to shift. Yes, that's correct. Yeah, because there is still the tardigrade, as you can see, is not, luckily, not completely crushed. And because of the fast movement, maybe you can see the claws a little bit. It's difficult to see because it's moving so quickly, but actually, at the end of the feet, there are some claws. Yeah. Okay. Tardigrades even live in oceans. Yep, underground streams. Yeah. Do you have phase contrast? Yes. Oh, yes, I do have a 40 times phase contrast. So if you want to, you can look at it. It doesn't look that great. Phase contrast really looks very good for very thin objects. But because you're asking, I'm just going to switch over to phase contrast, and so this one is now this one is now bright field. Okay, so that's basically how. It's the standard view, so as you would say, okay. Then DIC, yeah, you have to, which uses a lot of light, and yeah, if you adjust it properly, you're going to see that there is a slightly more contrast. And now let's do the following. I want to show you now phase contrast. I almost forgot about it, but yeah, it's not that great. And I have to change now the condenser as well. And I have to open it up, turn it down. Is this correct? No, that's the one. Yeah, so that is now phase contrast. Okay. Yeah. Yeah, it's a little too dark still. I can I'm now adjusting the camera, making it a little bit brighter here. Yeah, you see, phase contrast is very good for observing thin objects, but thicker objects like this one over here, they don't look so good, right? I actually would probably prefer bright field. So let's go back again to the other one, to bright, and close the condenser again a little bit to increase the depth of field. Yep, here we go. Yeah. Yeah. Depends. For some sample, it looks very nice, but for others, yes, exactly. I do not want to have any residue or dust and light path being captured through the camera. Yeah, I get it, but unfortunately, dust is a general problem. It's a general issue. What you can do, and this depends a little bit on whether it's really worth it. Some microscope companies actually do offer cleaning services as well. So basically, they will take it apart and do the cleaning for you. I've had this actually done a couple of years ago with a used microscope, a used Olympus microscope that I was almost unusable because it was already so old and dirty and so on. I paid 200 euros, sent it off to the certified Olympus service company. The guy was actually servicing microscopes for universities and so on. And I gave him also my microscope and said, please get it serviced. And he completely took it apart and got it back almost like new. So that is also possible, but completely taking apart a microscope is... I tried that once and I could not put it back together again. I'll be honest. The mechanical stage was so complicated that I would actually not risk it, unless it's really necessary. No one. Okay. The professional cleaning has to be done in clean or anti-static room. I don't know about that, whether it has to be done in an anti-static room, but I'm quite sure that that it's got to be. No, I don't think it's necessary to have it clean like in an environment like where you do make semiconductors, right? I think that's not necessary, but obviously it's got to be reasonably clean. I guess maybe they're also using some kind of compressed air, to remove the dust and so on. It gets me nervous when microscopy has to be taken, when microscopes have to be taken apart. Yeah, honestly, I tried it on Olympus, an old Olympus CHA microscope. I even had the instruction manuals, the service manual. Everything was documented on how to take the mechanical stage apart. I didn't have the right tools, I discovered, and the ball bearing system was so complicated that I really had problems putting it back together again. So ever since then I said don't touch a running system. Yeah, and if the microscope works reasonably well, yeah, then don't take it apart. I mean, taking the objective out and so on is not a problem. Yeah, but I'm talking here about the actual mechanics, the coarse and fine focus knob, the mechanical stage, all of these things. Yeah. Semiconductor companies do have their clean room. Okay, exactly. Yeah, I think for microscopes it's not that yeah, critical. But where is this again? Yeah, let's give it again a little bit of color. Okay, well, I have to tell you, I'm relieved that I was able to show you finally a tardigrade. Yeah, how these are there any mini tweezer-like tools to pick up and grab the tardigrades? You're talking about so-called micro manipulators, and essentially what you could do and what is possible is if you use a microliter pipette like the one I have over here. This is a 1 milliliter pipette, but there are also smaller ones. And I think the smaller ones are maybe more suitable. And what you can do is you can actually then go here on the slide, and you, with a second hand finger resting, yeah, on here, you can actually try to pick up and take up some water samples this way. Huh? So it is possible. I have done this before. It requires a very steady hand, and it's a little bit of question of luck as well. Yeah? And then I've done this in order to kind of remove all of the other dirt and so on. But in the process, sometimes you also lose the specimen. Yeah. So yeah, try using a UV flashlight on the target. That's interesting. Let me just a second. I'll check if I have a UV flashlight. How do I put this on? Okay. Yeah. Okay. I never tried this. So I've got some. I have to turn this off. So the request is check for fluorescence. So I'm going to do now the following. I'm going to simply turn off everything here, and I've got some UV flashlights here. I'm going to, and I don't know if this actually works. No, I don't see anything. So there is no autofluorescence, or it's so weak that I'm not able to see anything. Huh? So that's and there's another one here with a different frequency. It's a little bit more blue. Nah, not very much. Lower magnification. No, I guess, yeah, you have to actually shine it in from the bottom. Yeah, but this does not seem to be. Yeah. So what you have to do is with a UV light, you shine it in from the bottom, and then you use a dark field patch stop. So this basically means that the UV light will strike the specimen from the side. And then the fluorescent light is going to be emitted. So this actually does work, but sometimes you need a very long exposure, because sometimes the autofluorescence is very weak, and then you need to expose for a very long time. Okay, but I have tried this by actually putting some blue LEDs, and sometimes you get a very weak fluorescence. But I think shining it from the top, it might not be enough. Okay, that's a little bit the issue here. I'm going to try both of them with the different frequencies, or maybe, yeah. Nah, it's too dark. You know what I could try? I could try to make it much brighter. So I'm going to increase the exposure. Now even here, the camera is not able to detect anything. Which is kind of surprising because even when it looks through it, no. Because normally what you would expect is some sand or some material to kind of, yeah. Okay, now I accidentally bumped the... Yeah, here it is again. Here it is. Yeah, and now it's kind of, I have to go down again with the exposure time and up with the light so that the frame rate is again. No, that was interesting. Okay, thanks for the recommendation, but it's something where I need to make sure that the light comes from the bottom. Yeah. So, okay. I forgot where I left off here. Semiconductor, tweezer, UV flashlight. Here it is. Okay. Pipette transfer works great for tardigrades and rotifers. Yep. Okay. Tardigrade is running away. Yep. Here it is again. Yeah. I missed the scanning and transmission electron microscopy courses I took years ago when I took my biology degree. I can't afford to buy them for personal use, however. Yeah, look, now it's trying it. Now it got free from the how do you call it? The sand. Of course, electron microscopy is cool, but light microscopy has the advantage that you're able to see movement. Yeah, so that is a little bit yeah. But of course, the details of electron microscopy, I mean, that's of course fascinating as well. Yeah? But then again, yeah, I don't know if electron microscopy is so suitable as a hobby because of the elaborate preparation that's necessary. Now it's going off. Look at this. Let's see what it does now. Yep. Is it male or female? I wonder if they're hermaphrodites or not. Could it be that they're both? It's not unusual for some of these animals to be both. That could be one thing, but it could also be that they actually have two genders. It's yeah. You know what? I'll quickly Google that. Let me see. Where's my tardigrade? What does it say here? Hermaphroditism in Tardigrades, a publication from 1979. Evidence is presented to show the existence of hermaphroditism in tardigrades. So it's possible that apparently it's possible that they are both male and female at the same time. Oops, now it's gone again here. At least this is according to some research. Then again, with 1,300 species, yeah. The cheapest electron microscope I could get through a friend was about $12,000. Wow. Yep. But I think that the specimen preparation is quite elaborate because you need to completely dehydrate the specimen for electron microscopy. You also need to I think coat it in a layer of metal so that it doesn't burn up the specimen. So apparently, I've never worked with electron microscopes before. Yeah, and I think that I've darkly remember had a short email communication with someone who actually even collected electron microscopes. and then basically repaired them. Yeah. Yeah, folks, I've been now talking 1.5 hours again. I think it's time to, yeah. Can you freeze a tardigrade? Yes, they're very, tardigrades are very cold resistant. Not so much heat resistant, but quite cold resistant. Yeah, so it's actually something I might be trying. Suggestions is to dry them and to see if they're able to survive a week. Yeah? But I think I'm going to call it quits for today again. And yeah, at least we found a nice one here. And yeah, if you want to see more of these videos, then do check my other channel and search for tardigrades, because I think I made some years ago a few of them. Yeah. And I actually was able to, oh, look at this, yeah, a protozoan. Again, almost the same size as the tardigrade. Yeah. I was really stressed out at the beginning of the live stream, because this one tardigrade that I wanted to show you was actually gone, but we were very lucky that we were able to find one. Yeah. Check out government surplus days for bargains electron microscopes. Ah, yeah, yep. Used microscopes by universities, maybe. Yeah. Yeah, I'm going to call it quits as well after 1.5 hours. And see you again next week. I wonder what I'm going to talk about next week. I'll think again of another topic. Yeah, I've been recently doing a lot of direct microscopy work, but maybe I'm going to do a little bit of hardware again. Some years ago, I made a Lego microscope, a homemade Lego microscope, which actually works. I might show that one to you if you're interested. It's a little bit more of a fun project. Yeah, yeah, and I think I'm just going to call it quits, and I'm just gonna do it like this, so that it doesn't run away too quickly. Yeah, I think I'm going to say all the best to you. Have a nice week. Still have a nice remaining weekend, and yeah, happy microbe hunting. Happy microbe hunting, everyone. And yeah, and I've been always wondering: are tardigrades and rotifers, are they microorganisms or not? Well, they are microanimals, but because they're multicellular, strictly speaking, they don't classify as microorganisms, or do they? What do you think? Maybe you can leave that in the comments below, or some food for thought for the upcoming week. I wish you all the best in any case. Yeah, hope you liked it again, and see you around, okay? Yeah, all the best, and bye-bye.